Hello, my name is Ksenia and I'm an application specialist in BioVitrum company. Today we will continue talking about special stains in histology. Hematoxylin and eosin staining gives us an overview of the tissue structure, and different special stains can help us to identify different structures, pathological processes in tissue or microorganisms, which is important to diagnose different diseases. Tuberculosis is an infectious disease which usually affects lungs, but can also affect other parts of the body. It can be active or latent. Treatment from it requires multiple use of different antibiotics for a long period of time. Tuberculosis has been present in humans since antiquity, but still it is a huge problem all over the world. Roughly about 1/3 of the population of planet has been infected with tuberculosis. It is the second most common cause of death from infectious disease after HIV. The World Health Organization declared tuberculosis a global problem in 1992 and even developed a global plan to stop tuberculosis. But nevertheless, in 2015, there were registered about 10.4 million new cases of this disease, and about 1.4 million people died from it. The bacterium causing this disease, Mycobacterium tuberculosis, was identified and described in 1882 by Robert Koch. He received the Nobel Prize for this discovery in 1905. Mycobacterium tuberculosis is small, aerobic and non-motile. If the gram stain is performed on the clinical samples, then Mycobacterium either stains very weakly gram-positive or does not retain dye because of the high lipid and mycolic acid content in its cell wall. Since Mycobacterium retain dye even if it was treated with acidic solution, it is classified as acid-fast bacteria. The first and most common method to identify acid-fast bacteria in human tissues was introduced by two German doctors, Franz Ziehl and Ziehl-Neelsen. Nowadays, it is known as the Ziehl-Neelsen staining. Dr. Franz Ziehl was a German bacteriologist. He introduced carbofuchsin to stain tubercle bacillus in 1882. Ziehl-Neelsen was a pathologist. He studied medicine at the University of Leipzig, where he received his doctorate at the age of 22. Later, he became a professor at the Institute of Pathology of the University of Rostock. He died at the age of 44 in 1898, quite young, presumably due to pathogen exposure during many years of his bacteriological research. Together, Franz Ziehl and Ziehl-Neelsen developed a method for staining of acid-fast bacteria, and this method is used until nowadays in diagnosis of tuberculosis and to detect the presence of other mycobacteria. The reagents which are used in this method are carbofuchsin, acid alcohol, and methylene blue. Carbofuchsin is a mixture of phenol and basic fuchsin. It has an affinity to mycolic acids from bacteria cell wall. It is more soluble in these cell lipids than in acid alcohol. That's why the bacteria will retain the initial red color even after destaining in acid alcohol. Carbofuchsin is also used as an antiseptic. Methylene blue is the medication and a dye. As a dye, it is used as the component of many special stains. Here are the main steps of the staining protocol. After deparaffinization and rehydration, slides with thin sections are stained with carbofuchsin. Then they are washed in water and differentiated in acid alcohol. Then methylene blue is used as counterstaining agent. As a result, acid-fast bacteria will be bright red and all the background will be pale blue. What is important to remember when you apply a Ziehl-Neelsen stain? First of all, care should be taken to avoid over-counterstaining, as the scant organisms can be easily obscured. Secondly, decalcification with strong acid can destroy acid fastness of bacteria. Thus, formic acid is recommended to be used for decalcification. And besides, contrastaining agent can be substituted if color blindness causes the recognition problems. Nowadays, there is a wide variety of ready-to-use staining kits from different manufacturers. If you follow the protocol given by the manufacturer, you will be able to achieve good results of staining. Concentrations of the solutions and time of exposition is chosen to get fine color scheme. If this video was useful for you, press like. If you have any questions or something to add to this topic, please write in comments and subscribe not to miss our new videos. Thank you. See you soon.